relatively new group of plant hormones. Okay. So basically, in Australia, there was a very much frequent phenomena of fire forest. As recently in California, there was a big calamity and huge properties were lost by the fire forest. So similarly, in Australia, that was a common phenomena. In the jungles of Australia, the fire forest was a common phenomena. So the scientists examined that after fire forest, in the next season, the growth of plants was very much abundant. So they found out that by charring and burning of leaves and twigs and wood of the jungles, some organic compounds were released by this smoke, and these compounds were very much effective in increasing the growth and particularly the seed germination. So this work was first started by some scientists of Australia. So later on, much work was done on karrikinolides, and now it has been established perfectly that karrikins, which are found in smoke water or smoke, these compounds are very much influential in enhancing the growth of plants and particularly seed germination. So by stimulating with this research, I performed this work and I designed an equipment for generation of smoke water myself. So as I have told you that karrikins were discovered by some Australian scientists, and they found out that after the fire fast, when the rainy season come, after rainy season there is a huge growth of plants because those smoke compounds were retained in the soil, and soil is also a bank of seeds, a good source of seeds. So those karrikins stimulated the seed germination, and the growth of the plant was very much luxurious, and abundant growth occurred due to the action of the compound which is found in the smoke of burning of plants or leaves. So this is Australia and New Zealand. So in both places, this phenomena happened, particularly in the Western Australia. Western Australia, this invention was made. This is the Western Australia, this one. Perth is here, Perth, and this all. So the scientists of University of Western Australia, they did this research and found out that smoke of plant can stimulate the growth and germination of seed. Later on, it was found out that the new group of compounds, karrikins, they are very much effective in causing the seed germination and general growth of the plants. So this word karrikin comes from Australian language, which means smoke. In fact, so this is the slide of fire forest in Australia, and most of them are eucalyptus trees. And Australia is very much famous for eucalyptus trees. You will be surprised to know that in Australia there are 900 species of eucalyptus. Right? I have also seen, I have visited Australia 2 or 3 times and found out a variety of eucalyptus. Okay? Not only tall plant, but sometimes the plants are like people and bargad and expanding, and not very tall. So Australia is very much famous for eucalyptus. So by the burning of the leaves and twigs and wood, these karrikins were produced. So karrikin role in post-fire vegetation regeneration. Slide A is the blaze of blaze produces smoke and ash with karrikin. This slide here is by the blaze and the smoke is produced. Number 2 slide shows after the fire, karrikins appear on the earth surface. After rainfall, the karrikins which were dissolved in the air, they come to the ground on the earth, okay, and they are stored in the ground like seeds. In the third slide, following the first rain, karrikins increase soil seed bank, germination and new plant growth. You see, the third slide shows the luxurious growth of new plants and high rate of seed germination occurred. So this was happened, and what led to the discovery of karrikins? For many years, it was recognized that the chemicals that a fire produced, rather than the heat, encourage the germination of some fire flower seeds. Both smoke and water that was drenched in chair or that smoke was passed through creating smoke water may have the same effect as fire. So, to find the active ingredient, several teams of scientists worldwide separated the hundreds of compounds in smoke water into distinct fractions using liquid chromatography and then tested each fraction for seed germination activity. The active fractions underwent additional fractions analysis. This investigation ultimately isolated an active molecule and chemical synthesis verified this finding by Filamenti et al. This compound contains a specific type of electron known as butanolide fused to a pyran ring with the systematic name 2-methyl-2-hydrofluoro-2,3-pyran-2-one. Subsequently, several closely related compounds were discovered in the smoke and collectively called as karrikins. These are called as karrikins or karrikinolide. Both are synonyms. So, how this word was used, I will come to this slide, that slide later on. So, in fact, karrikin in Aborigines language is called to smoke. Smoke को ये लोग karrikin कहते थे. In fact, in Australia, the original natives were Aborigines people. Aborigines, okay. When the European people went over there, they genocided the original natives of Australia, which are Aborigines, and they killed them. And you will be surprised to know the government used to issue the license to kill the Aborigines. They have only sword or arrows, but these Europeans, they have guns, so they killed them, and many of them vanished together. I have also seen Aborigines over there. So, so in Aborigines language, karrikin means smoke. So this karrikin word was derived from Aborigines language, which Aborigines people used to say. So therefore, these compounds are called as karrikins because they are derived from karrikins, from smoke, and the Aborigines people called the smoke as karrikins, right? So this is a slide of Aborigines people, the natives of Australia, and still there are some tribes are there. They are surviving there, but they live far from the population and they live in the jungles. So these people called as karrik to smoke. Therefore, the name of compound is karrikins. So there are many type of karrikins, KR1, KR2, KR3, KR4. Many compounds are there with little difference. For example, KR1. Okay, this is the most important karrikinolide or karrikin, which is commonly found in plant smoke. KR1. KR2 is same, but one methyl group is missing over here. KR3 is the same, but one additional methyl group is attached over here. KR4 is same, but here the additional methyl group is attached over here. Similarly, these karrikinolide can also be obtained by the burning of sugar. If you burn the xylose sugar, you can also obtain karrikin 1 and also estragol. So astragalactones and karrikins are very close to in their function, and their astragalactone is also a good plant hormone. So the Karrikin family, the first Karrikin discovered was KR1, also known as karricolite. Since Karrikins can be produced by burning sugars such as xylose, the pyron ring of Karrikins is probably derived from such pyranose sugar. Both Karrikins and astragalactones hormones such as estragol have a butylide ring. So these are the people of Aborigines. Karrikins are produced by the fire. It was discovered that the burning of many various plant products, including straw, cellulose, filter paper, and even sugars, could produce karrikins. Their synthesis from polysaccharides and sugars explain the pyron ring of karrikins, which is thought to be formed directly from pyranose sugars found in plant material. Karrikins are most likely produced at home by toasting and roasting specific plant foods. Cigarette smoke enhances seed germination, most likely because it contains karrikins. Further research has revealed that karrikins become unstable at extremely high temperatures. So this pyron ring is a common feature in the karrikins, the hexocyclic compound, and having oxygen in its heterocyclic ring. Karrikins also produced by some factories, for example, sugar factories. In sugar factories, the bagasse, उसका जो खोजड़ होता है, यानी एक्सट्रैक्ट निकालने के बाद में, that is known as bagasse. So bagasse is used as a fuel. Okay, when these factories are started, first they use some firewood, but later on, when this bagasse is produced in huge quantity, then these bagasse is dried in sun and used as a fuel. So by burning out these bagasse, karrikins are also produced in the environment. And after rain, the karrikins come to the soil, which can increase germination of the existing seeds. So, so I also worked on karrikin, and I designed a apparatus which can produce the smoke water which contains a lot of karrikins. So I worked on Daucoscarota, and then Trigonella funiculum, that is methi, then Corynus sativum, dhania, and then Anthem gradans, soya, and fennel. So we worked on 5 plants, and purpose of study was because lack of arable land, the arable land is insufficient. Secondly, increasing the demand of global market of the plant. Thirdly, that to increase the turnover. And fourthly, genetic potential of the plants was realized on the maximum by the fertilizers. So we can't increase the productivity by application of fertilizer because maximum genetic potential has been realized. So to overcome these problems, we worked on this smoke water plant, and this smoke water and karrikin can break the dormancy, stimulate the seed germination, enhance the growth and productivity, help in uptake of nutrient following somatic embryogenesis, displays activity at a very low concentration, interactions with endogenous plant growth regulators, no mutagenic or genotoxic effects are reported. So there are 71 compounds reported till date, out of which karrikins and cyanohydrin are the most important, which can stimulate the seed germination. So I have told you that there are different types of karrikins, KR1, KR2, KR3, KR4, KR5, and KR6. So I have done on KR1. So this chemical was purchased by a Canadian company, Toronto Chemical Company, and you will be surprised to know that 1 milligram cost of this KR1 was 25,000 rupees, more expensive than gold. So I purchased 1 milligram from Alberta Chemical Company, and then for the sake of comparison, because if you don't have a standard, how can you compare? So therefore, we were compelled to purchase this 1 milligram of KAR1 from Canada, and then we worked on smoke plants. Okay, so this apparatus was prepared by myself, and in this there is a hot plate which was purchased from a Mumbai company, and then hot plate, on the top of hot plate there is a petri dish which contains the straw, because straw, burning of straw produces Karrikins. Okay, so we took the straw, clean it, and then dried it. After drying, it was put in the petri dish, and then the hot plate was turned on. By the heat, the straw was charred, was burnt into smoke, and this smoke was passed through this tube. Here is a suction flask. Okay, in suction flask we took the distilled water, DDW. Okay, and then the outlet of the suction flask was connected to a motor. I purchased this pump from USA. That was not very expensive in those days. So I used this pump, suction pump, and then the suction pump was turned on. So this smoke path was like this. It was generated by the straw, then went to this tube, and from this tube it was dissolved in water. By the suction of the air, there was bubbling in the flask. Okay? By the huge bubbling, the smoke was dissolved in water, the DW, right? But a point came when there was no dissolution of smoke, so saturation point came, and this water was called as SSW, smoke saturated water. So this smoke saturated water, SSW, was used in our study, okay, right? So after each 5 minutes, we also read the optical density of this smoke water, okay? And after about half an hour, the optical density remained constant, and this showed that no more smoke is going to be dissolved in water, and now the water is fully saturated with smoke. So this is called as SSW, and then SSW, smoke saturated water was diluted to 1000 times, 500 times, 100 times, and 250 times. And this diluted smoke saturated water was sprayed on different plants, okay, which stimulated the growth and yield of the plants, right? Shari Sahab, can you run this small video? I have also a small video of this. So you can see that the hot plate is working. Temperature goes to 500 to 600 degrees centigrade, and at this temperature the charring and burning of the straw starts. Smoke is produced, and this smoke passes through the distilled water because the air is sucked by the suction pump. Okay, we can regulate the speed of suction by a regulator, which is I will show you. Uh, by this regulator, we can control the speed of supply of smoke. This is the regulator. By this regulator, we can control the speed of bubbling or the speed of smoke flow in the water. Okay, and as I have told you that after 5 minutes, each 5 minutes, we just took a micropipette, take a sample of 0.5 ml, and read the optical density using a spectrophotometer, right? So optical density was increasing, and after 35 minutes there was no increase in the optical density. It showed clearly that no smoke is dissolving in water, that is, water is fully saturated with smoke, right? Then the machine was stopped, and the smoke water was taken, and it was diluted to 500 times, 1 to 500, 1 to 100 times, 1000 times, and 250 times, right? So we found out that dilution factor is 1 is to 500 was most effective in obtaining the maximum seed germination and in obtaining maximum growth and yield of the plant. Okay. Okay, now we come back again to the PPT. So the whole apparatus was prepared by me, and this paper was published in Journal of Plant Growth Regulation in those days. I think about 6-7 years ago. So this is the smoke saturated water, which was filtered by Wattman filter paper. Yeah, this is the main graph. You see, about 32 minutes, the water was fully saturated with smoke. So this water is called as SSW, smoke saturated water, which was used for our study, and this water was diluted to different dilutions and was sprayed on plants. Okay, so we found out that with the dilution factor 1:500 was the most effective in increasing the growth and yield of plants. Even the active concentrate was also maximum at this concentration. Okay. So this work was a part of PhD program supervised by me to one of my past students, Ms. Arshia Aqil. Arshia is currently pursuing her postdoctoral research program in Copenhagen, Denmark. Nowadays she is in Denmark and doing some molecular work in Denmark as a postdoctoral fellow. So we separated this smoke water by Wattman filter paper, and then HPLC was run. HPLC was run, and we found different peaks of compounds, and then we are also very much thankful to Professor Javed Iqbal Sahab in our chemistry department. Now he is no more with us, but he was a very dedicated person. He spared days and nights to recognize that which compound is KR1. Fortunately, by using a compound library, he was successful that this compound 153 peak number was the KAR1. Okay, so in this way he was successful to identify that this peak number 153 was the peak of KAR1, which is very small amount. KR1 is found in the smoke water. So in this way, we use different dilutions of smoke water. You can see 1:1000, and 1:500, 1:250, and 1:100. And this is the control, that is the double distilled water. So similarly, we also sprayed the Karrikins compound, which we were purchased from Canada, Alberta University, Alberta Chemical Company, and 4 concentrations we took: K1, that is 10 power minus 10, K2 10 power minus 9, 10 K3 10 power 8, K4 10 power minus 7. So 5 concentrations, including one control, we took from the smoke water, and 5 concentrations we took from the Karrikins, which we purchased from Canada. Okay, 1 is control and 4 concentrations were taken. So we found out that 10 to the power minus 8 concentration, and in some cases 10 to the power minus 9 concentration of Karrikins, and in case of smoke water 1:500 in most of the cases, and occasionally in 1:250 ratio of smoke water proved best for the increasing the growth, yield, and quality of plants, and also the seed germination. So we performed many experiments on different plants, and in 5 plants we took 2 sets, 1 set for smoke water and 1 set for Karrikins, right? Smoke water, Karrikins, smoke water, Karrikins. In this way 10 experiments were performed on 5 plants. Clear? So we performed 10 experiments, and for each plant we took 2 sets of experiments. 1 set was designed for smoke saturated water, dilute factors, and 1 set was carried out for the Karrikins as in for the sake of comparison. So in a nutshell, we found out that smoke water was most effective in comparison to Karrikin alone was applied to the plant, because smoke water contains so many Karrikins. Therefore, obviously, its effect was most obvious and clear in comparison to KR1. But as far as the control is concerned, KR1 10 power minus 8 or 10 power minus 9 gave the maximum performance of the plant, including seed germination also affected. We studied many parameters of these plants, many photosynthetic parameters and phenol content, DPPH activity, and yield parameters, and seed germination parameter, etc. So in this way, we found out that the out of SSW, 1:500 dilution gave the maximum germination, you can see, right? In case of Karrikin, 10 power minus 8 gave the maximum germination value in case of 10 power minus 8 molar concentration of Karrikin. Similarly, chlorophyll content and other parameters, they gave the highest value at 1:500 in case of smoke sage water, and in case of Karrikin, 10 power minus 9 or 10 power minus 8 gave the maximum value. Plant weight, root length, diameter, fresh weight, root fresh weight. Similarly, for photosynthetic parameters also gave the maximum value in case of 10 power minus 9 minus 8 molar, and in case of smoke water 1:500 dilution was the best. So different parameters were found best by the application of smoke water as well as the Karrikin. So the trigonella, that is methi, contains a compound known as trigoline. Trigoline content was also increased by the application of Karrikins and smoke water. Okay, for example, here 2.6 milligram in control and 3.17 milligram per gram in case of they are treated by KR1 by smoke water. Similarly, by the application of KR1, it was 2.6 in control and 3.1 milligram in case of Karrikin. So, how does Karrikin works? This is still a question, but many of the steps have been discovered, and according to this, this is the action of the Karrikin and this is the action of strigolectons. Because both actions are more or less similar, therefore both are studied here. So this is the application of Karrikin and the application of strigolectons. In this, this step is still not discovered that what precursors are involved in the formation of these receptors, in which one is ubiquinone is very much common to both of the compounds. In this way, both the compounds they produce some secondary messengers. Okay, in case of Karrikin, it is like this. In case of strigolectons, it is like this. These secondary messengers go to the DNA and then they stimulate the DNA replication, and in this way the proteins are produced. Those proteins either they stimulate seed germination, or they induce the root system architecture, or they induce the mycorrhizal symbiosis. So in this way, this is the mechanism how Karrikins stimulate the seed germination and growth of the plants. So now, as my title of my talk is not only confined to Karrikins, I want to tell you that we have also worked on some other plant growth promoters, and now it comes a new technique of promoting plants by RPPs. RPPs mean radiation processed polysaccharides. Okay, so this is another group on which we worked about 2 decades, and we were granted 2 major projects by the Bioatomics Center. And 2006 they gave the first project on mint using RPPs, and the results were very encouraging. Then they said, now you should perform the experiment in the field. Okay, the first project was concerned with the pot experiment. When the results were encouraging, they asked us to do the field. Then we went to Sambhal, because Sambhal is a land of mint, because there in the agroclimate is very much suitable. Okay, therefore the BRC people asked that you should perform this experiment in that area where the mentha is grown very luxuriously and successfully. Therefore, we went to Sambhal, we land a piece of land of 7 acres, and we performed field experiment over there. So basically, there are some polysaccharides, particularly marine polysaccharides like carrageenan, alginate. These are found in seaweeds. Okay, seaweeds produce lot of polysaccharide like sodium alginate, carrageenan, okay, and so many. So when these separated or extracted polysaccharides are subjected to gamma radiations, these polysaccharides are broken down into smaller molecules. ठीक है ना, जैसे आप किसी शॉपकीपर पास जाएं कि भाई ज़रा 1000 का चेंज दे दो, तो वो 500 का, 100 का, 200 का नोट दे देगा आपको. Similarly, by the action of gamma rays, the large molecules are broken down, they are decomposed to smaller molecules. These water-soluble smaller molecules, when applied to the plant, they can increase the growth and yield of the plant. Okay? So initially this work was done in some like Manila and Philippine countries. So then I submitted a project to BRC that if we apply this technique to medicinal plant, can we increase their yield and quality? So they appreciated this idea very much, and they gave this project. So we worked on sodium alginate, carrageenan, and chitosan. Chitosan is also a polysaccharide, but this is an animal-derived polysaccharide, which comes from crabs and shrimps. Okay? समंदर किनारे जब waves आती हैं, so they bring the small bodies of these shrimps and prawns and crabs. So when the waves go back, the laborers pick those dead bodies of shrimps and prawn, and they collect in the sacks. Okay, then these are taken to the factories. In factories, they are broken down into small pieces and then boiled. Okay, in the boiler. After boiling, the chitosan is extracted. Okay, now this chitosan is purchased by Sigma companies like that, and then we purchase it from Sigma company. The chitosan then we send to Bhabha Atomic Center. In Bhabha Atomic Center, they expose the chitosan to the low dose of gamma radiation, okay, about 200 kilogray or so. In this way, the large molecules were broken down, depolymerized to smaller molecules, right? So when these smaller molecules are dissolved in water and sprayed on the plant, they increase the growth, yield, and quality of the plant. Okay, alginate and carrageenan are water soluble, but however, chitosan is not water soluble. You have to use some acetic acid to dissolve it into water. Okay, so we dissolve this radiation process polysaccharide into 0.3% acetic acid solution, and then it was successfully dissolved. Then we sprayed these solution on the plant, and we found very useful and very spectacular results of these polysaccharides, right? You got the point? So we did not use radiation on the plant directly. We realized the benefit of radiation indirectly. Okay? We did not expose the plant to the radiation. Rather, we use radiation technique to only break down the larger molecules into minute molecules, right? So in this way, there was no effect of radiation on the plants, or the product of this plant will not harm any human being which is going to consume it, right? So this is totally safe from radiation effect. We use radiation only to break the big molecules into smaller ones, right? So we use this work on mint, mentha, and mentha contains a lot of menthol, which is used to prepare balms and wigs and many cough syrups, okay, which is very much effective in making cough syrup, like, okay. So we have collaboration with BARC, and Dr. Lalit Varshini was my principal collaborator, and he was a good scientist. His base was chemistry, and then we worked on this project and we found very interesting results on on radiation process polysaccharide. So in this way, we worked. So I would like to tell you that these large molecules can also be broken down by different methods. For example, you can use the bacteria, or you can use other technique. But this technique, by breaking the molecules by radiation, is very much safe and quick and very successful. Okay, you can break these polysaccharide by gamma rays or electron beams. Okay, and in this way we can found these smaller molecules which show antimicrobial activity, which also show illicit activity in plant, promotion of growth, suppression of heavy metal stress. So these compounds are very useful in solving many agriculture issues. So first is sodium alginate. So we also did the GPC of this compound, and then is chitosan. As I told you, that this is an animal product, chitosan, but you should know that effect of chitosan on plants was very much overwhelming than the carrageenan and sodium alginate, right? So it was dissolved in acetic acid and then it was sprayed on the plant. So different plants were chalked out, and then we sprayed these chemicals on plant, and then we were successful in increasing different parameters like growth parameters, biochemical parameters, yield parameters were increased. This is the box of sodium alginate, and this is the sodium alginate in a petri dish. So these molecules are very large in size. You see, only one unit has been shown, but thousands of units are there. By the action of radiation, these long chains are broken down to smaller molecules, which are known as radiation process polysaccharides, or you can also call them oligosaccharides. Oligosaccharides mean smaller broken molecules. So these smaller broken molecules were dissolved in water or acetic acid and then sprayed on the plant. This is gamma radiator in BRC, where these chemicals were broken down into smaller molecules. Okay, these are the images of electromograph before breaking and after breaking, right? So then these chemicals were dissolved either in water or acetic acid and then sprayed on the plant. So this project we performed at Sambal, about 200 kilometers away from here in the north direction, and this area is very much famous for mint production, right? And then this was a project run on the mint. This is my student Hassan Jalil from Jammu, okay, in this project. Then the chemical was sprayed by using electric spray machine. The machine was charged at night, and the daytime the sprays were made on the plant. You can see very huge and very luxuriant growth of the plants were happened. The nearby farmers were also surprised to see that, sir, your plants are very much healthy than our plant, and moreover in your plant there is no insect attack, but our plants are also affected by insects. Okay, so we can obtain two benefits. Number one, high growth, and these chemicals were also used as pesticides. Okay, so both the benefits were obtained. This was project on mint. You see control plant gave this much oil, and then 40 ppm gave this much oil, and this 60 ppm gave more oil, and 80 ppm gave the maximum oil production in case of chitosan application. So then the oil was run for GCMS, and the fractions were separated and quantified by GCMS machine. This GCMS is also sanctioned by the BRC project. We purchased so many equipment by this project. GCMS, spectrophotometer, and so many smaller equipment. So in this way, uh, for uh, we perform this experiment in the Sambal in 3 types of plots. Okay, 10 square meters, 100 square meters, and then 1000 square meters. Because bar people said that you should do this experiment in the field and you should increase the site so that we can confirm that the results are reliable and valid. So therefore, for 100 square meter plot, we purchased this equipment from CIMAB Lucknow, and in this way, ये back कैसे होता है? एक slide, हाँ. Yes. So this distillation plant was for 15 kg of biomass. You can put 15 kg leaves of mint in this container, and this was run by the LPG gas. Okay. After 5 hours distillation, the oil was extracted from the leaves of mint. Okay. So, but for the last experiment which we perform on 1000 square meters area, the biomass very much huge. Then we harvested the whole biomass from the field of 1000 square area, and then we carried out this biomass to the mint factories, okay, menthol factories. The whole biomass was dropped in a very huge container. Okay, these huge containers are filled with this biomass, and then the lid was covered, water was filled, and run for 5 hours. The oil was extracted by this technique. And you will be surprised to know, in case of control plant, we obtained only 4.73 kg per 1000 square meter area of the land, but in case of treated plant, we find 7.56 kg in 1000 square meter area. Okay, so in this way, about 60% increase was obtained. I represented by report at BRC, and all the scientists were very much happy. And Dr. Vashni is also working that how to get it patented, this technique. Okay. So not only the oil content, but also menthol content and essential content was also increased by this treatment. So after Karrikins and after RPPs, we also work on tricontrol. This is a long chain alcohol. This is also growth promoter available in the market. Okay, this has also a miraculous effect, and we did a lot of plant. And then the fourth plant growth is new one. This is known as MDM, methyl alpha D manoperoxide. So this company is in USA. I wrote a letter to them that I am working on medicinal plant. None of you you applied this chemical on medicinal plant, so we want to check it, that whether this chemical can also work on medicinal plant. So they made a meeting and they granted 1 gallon of this MDM and sent to us. They say that by the application of MDM, the cherry yield can be increased by 10%. Okay, this is the company name, branded Hammer. And in case of watermelon, 26% increase was done. In case of tomato, 21% increase. Okay. In case of cantaloupe, खरबूजा जिसे कहते हैं, 12% increase was done. In case of lettuce, salad, 24% increase was done. In case of pepper, Shimla mirch, 11% increase was done. So this was the report by the branded Hammer. So then I wrote to them that if we apply this chemical on medicinal plant, can we increase the growth and yield and quantity of plant? So they were happy and they granted a gallon of this chemical. Blueberry, ये बहुत full of antioxidant, बहुत ज़्यादा इसमें interaction होते हैं, and very expensive also, 26% increase. In case of cabbage, 7% increase was done. So then they gave this MDM to us, and the mechanism of action of this is that when the Calvin cycle is over, millions of glucose molecules are synthesized, as you know very well, right? So many of the glucose molecules are trapped by plant proteins known as lectins. Okay, lectins trap thousands of glucose molecules, and when those glucose molecules are trapped, they are useless for plant. They can't help plant in their growth and yield. So what do they say? That their chemical, MDM goes to the lectin where the glucose molecules are bound, and the MDM says that get out from here. So the MDM is fixed over here, which is a non-toxic compound, and the glucose molecules are released. When these glucose molecules are released in the plant, they are utilized in different metabolic phenomena like respiration and this and that. Okay, so this is the concept, their concept, that MDM releases the glucose molecules which are trapped by plant lectins, plant proteins, and when the glucose molecules are released in cytoplasm, this is utilized in many constructional work in the plant cell, right? So we did this work on curcuma longa. हमने हल्दी पे काम किया इस पे यही डिपार्टमेंट में, and we found out that fresh weight, dry weight, chlorophyll A, chlorophyll B, carotenoid, electron transport rate, phenol content, flavonoid content, and leaf thickness, stem diameter, curcumin content was also increased by the application of this new chemical, new PGR, known as MDM methyl monoside. So they said that this chemical is harmless, and this releases the glucose molecules which are unfortunately trapped by the plant proteins lectins, and when these glucose molecules are released from the plant proteins, okay, they are utilized in different metabolic phenomena in the plant, and in this way this can increase the plant productivity. So in this way we did a number of work on different plant. So this is the paper which published on Karrikins. I have also brought some copies of this paper, and you can take one copy for each of you. आप लेक्चर के बाद यहाँ रखे हुए हैं, एक एक कॉपी ले लीजिएगा, ठीक है? Okay. Okay. Now this is over. If you want to ask anything, I will try my best to reply, whatever I know. If you want to give any suggestion,